So have you ready? Can I start? Can I start the lesson now? Okay, thank you. Last week, we talked about the digestion process from mouth until stomach. So now you already remember that stomach, they are playing very important roles in the protein's digestion because they contain the pepsin enzyme, and also they have the hydrochloric acid, help them to create the acidic environment for the optimum activity of the pepsin. So today we are going to learn about the digestion process in the small intestine. So now the food already come out from the stomach, so they reach the small intestine, which is here. Okay, so the small intestine are divided into three parts. Generally, they are divided in three parts. The three parts we know as the first part is duodenum. I think you know it. Duodenum. Then secondly will be the jejunum, and then ileum. First part duodenum. Second part is jejunum, and then third part is ileum. So those are the three parts in the small intestine. So duodenum will be the first part which is they receive the acidic chyme. Okay, chyme means what? Chyme refer to the food state with the semi-solid. We call it chyme from the stomach. Okay, so this is the duodenum. As you can see there, they receive the content from the stomach. Okay, so now we talk about the digestion process in the duodenum. So the duodenum is a process which is the digestions will be happen, but please remember that duodenum will not secrete any enzyme, but they will receive the enzyme. Okay, this is the enzyme. Pancreatic amylase, trypsin, and lipase. Those enzymes are being produced from the pancreas. Okay, so those three enzymes are produced from pancreas. Pancreas is the organ at here. This is the pancreas. So pancreas, they will secrete out the enzyme, but the digestion process will not happen in the pancreas because the pancreas, they only release the enzyme, okay, through the pancreatic duct. Okay, so the enzyme will go from the pancreas through the pancreatic duct until they reach the duodenum. So you can see this is the pancreatic duct. Pancreatic duct is just like we call it as just like a small tube connected between the pancreas and the duodenum. So those are three enzymes produced by the pancreas. Okay, so the pancreatic amylase, they help to break down the starch, become the maltose. Then how about trypsin? Trypsin is another protein enzyme. Okay, because trypsin also help in the protein digestion, but now trypsin, they will break down the proteins, become the peptide here. So this is the equation of the trypsin. Polypeptide plus water converted into the peptide. And then lipase. Lipase also, they will break down the lipid, become the fatty acid and glycerol. And then, secondly, you need to know about the bile. Okay, because that bile is produced by the liver. See, liver is the organ, they will produce a bile. However, the bile will store in the gallbladder. Gallbladder will be another organ attached to the liver. Is that here? This is the gallbladder. So whenever the liver produces bile, the bile will store in the gallbladder. I mean the bile. Okay, the bile. So after that, whenever the duodenum require the bile, so the bile will be, transferred from the gallbladder through the bile duct until they will reach the duodenum. So you should know about what contents inside the bile. So the bile is actually alkaline. It's an alkaline mixture. Okay, because they contain what? They contain this sodium bicarbonate and the bile salt. So both components are alkaline. So the bile, the functions of the bile is include two functions, okay? Two functions you should know. The first one is the bile able to perform what? Able to perform the emulsifications. This one, emulsification. So what is meant by emulsification? Emulsification is the process which is they will trying to break down the fats. Okay, the fats initially will be like this, a very large lipid droplet, right? So they will, after they undergo the emulsifications, they will break down, become the very small lipid droplet, like this. So why they should break down the size of the lipid droplet become in this way? Because the purpose of this is they want to increase the total surface area. To increase the total surface area for digestions by the lipase. As you know that if the lipid droplet becomes smaller, that means that they have a higher contact. I mean, they have a higher area of contact with the enzyme lipase. So they're able to increase the rate of the enzyme digestions by the lipase. Yeah, so this is the importance. I mean, yeah. And then secondly, they're able to neutralize the acidic This one, neutralize. Because when the stomach contents coming from the stomach until they reach the duodenums, the content is acidic. The content is acidic. However, in the intestine, the environment should be slightly alkaline to neutral. So the bile, they will help to neutralize the acidic chyme. So initially, the acidic chyme, the contents will be about pH 2. For example, pH 2 or pH 3, which is very highly acidic, right? So after they will be neutralized by the bile, so the bile, they will be converts the pH become maybe pH 7 to 8. This is in the duodenum. So this will, I mean, the bile, they will help to provide an alkaline conditions for the enzymatic reaction, because those reactions by the pancreatic enzyme, they are actually preferred in the slightly alkaline environments, which is about pH 8, 7, 8 to 9, around that pH. Means if the pH become too acidic, it will be not suitable for those enzymes to take their actions. Okay, so most important that know the function of bile, which is the emulsification and neutralizations of the acidic chyme, and then you should know the three types of the pancreatic enzyme, okay, which is secreted by the pancreas, but they are acting in the small intestine. They perform the digestions in the small intestine. Okay, please remember about this equation, and then please remember to add the water in front of the equation, the water. This one, water. So those equations, they are known as the hydrolysis, which involve in the water to split the large insoluble molecule become soluble small molecule. And then I will talk about in ileum. Ileum is the last part of the small intestine. Okay, in the ileum, they will secrete the enzyme. See, the ileum contains the intestinal gland, so they will help to secrete the intestinal juice. So those intestinal juice, they will contain the digestive enzyme to complete the digestion of the peptide and the disaccharide. So for example, you have a look on this. Okay, iriopsin. Iriopsin will be the third protein that you learn. So these iriopsins, they will help to actually hydrolyze the peptide, become the smallest form, which is the amino acid. Okay, so you can see that how protein is being digested, because initially that you can see from here, proteins is a very large, complex, insoluble molecule, right? Proteins. So from the proteins, they will be break down by the pepsin, become the polypeptide change, which is from this structure become this change. So after that, the polypeptide change, they will be further break down by the trypsin in the small intestine, which is this one. So this polypeptide change which is in this way. After they undergo the digestion by the trypsin, they will become peptide. Peptide, it will be in this form, which is a shorter change. Then peptide will be further break down by their peptide bond. This is a peptide bond, like this. So they will release their free amino acid by the epsin. So in the ileums, they will also secrete the maltase. Maltase, they will help to break down the maltose, become the glucose. See this equation, maltose plus waters by the maltase, they become the glucose plus glucose. Maltose is the disaccharide, right? It's a disaccharide. Glucose is a monosaccharide, which is they contain only one unit. So after that, glucose will be absorbed by the intestine villi. And then how about lactase? Lactase is a kind of enzyme. They help to break down the lactose, become glucose and galactose. Glucose and galactose are the monosaccharide also. Lactose is also example of disaccharide. And also the sucrase. Sucrase, they will break down the sucrose, which is a disaccharide, become the monosaccharide, which is a glucose and fructose. And you can notice that the enzyme, there will be N in these words, ACE, ACE, ACE. Okay, so whenever you look at this word, you know that it's referred to enzyme and lipase. Okay, so those are the process of digestion. Okay, so now we get back to the exam note, yeah, for this one. Okay, please open this note again. So now you already know about the whole process of the food breakdown, starting from the ingestions, digestions, absorptions, and then now we look about the assimilations later. Okay, so today we learn about assimilations. Okay, so this is just now that I told you. Okay, you can have a read later. Just need to know that stomach is always acidic due to the presence of hydrochloric acid. And then how about small intestine? Small intestine is actually slightly alkaline, which is around 8 to 9. So that's why they require the bile. Okay, they require the bile. Okay, help them to neutralize the acidic kind. And then for your informations, you need to know that Ethan Suyang, I ask you questions. Waters, okay? Water can be absorbed by the intestine in small intestine and large intestine. But maybe you can guess, small intestine absorb more water or large intestine absorb more water? Which one? Water. I refer to the water. Intestine will absorb the water.
Large intestine.
Large intestine? The answer is small intestine. Actually, small intestine absorb more water than large intestine. Seriously. Okay, maybe you can take a note. Small intestine absorb. Actually, this is not written down in your notes or any of the book, but this come out in exam before. Actually, the answer is small intestine. Small intestine absorb more water compared to large intestine. Because the length of the small intestine is much more longer than large intestine. When you look at the structure, you will realize that, see, the small intestine, their length is much more longer than the large intestine. So that's why they are able to absorb more water. Okay, so for the large intestine, the function is they will not really carry out digestions. Whenever I talk about large intestine, I refer to the structure, which is we call it as a colon, rectum, and anus. You can have a look. Colon is at here, around here. Okay, colon. You heard about colon cancer, right? Usually they will have a cancer at colon area, colons and rectums, and also anus. So this three part is classified under the large intestine. colon, rectum, and the anus. So the large intestine, the function is they will just help to absorb water and the mineral ions. And in the rectums, okay, so during the when the food pass along the large intestine, they will absorb the water from the foods. So the feces become drier and drier. So until they will form a bug. So until the feces will be reaching the rectum, and then they will store the feces in the rectum. So rectum actually is at here. It's a very, very short passageway of the intestine, help to store some feces. And then the feces, they will expel through the anus. Yeah, the anus. Okay, the pancreas, yeah. They will secrete the enzyme in the alkaline fluid to the duodenum. So the function is, their aim is they will slightly increase the pH of the food coming out from the stomach, become more alkaline. Then how about the liver? Okay, liver, you already know that liver produce the bile for the emulsification and the neutralizations. But liver also, they will help in the, we call as the deaminations. Deaminations. What is meant by deamination? Deaminations, you will learn about today, is when your body unable to store excess proteins. Okay, you can see this. Amino acids are not used to make proteins. They will break down by the deamination to produce the urea. So later we'll talk about this. Okay. Okay, now we talk about the diarrhea. I think you experienced diarrhea before, right? Diarrhea is the conditions which is the feces is appear in the watery condition, expelled from the anus. See, loss of the watery feces from the anus. This is diarrhea. So diarrhea usually correlated with some kind of disease like the Vibrio cholerae. You heard about it, right? Vibrio cholerae is a kind of disease causing by bacterium. See this one. This is example of Vibrio cholerae. Vibrio cholerae. It's a kind of bacteria. So what Vibrio cholerae do? Vibrio cholerae, they are usually ingested by the contaminated food or water. Okay, usually by the contaminated food or water. So this is their route of how you get in contact with this Vibrio cholerae, ingested with the contaminated food and water. Usually it's from water. Water is which is already polluted. So this bacteria, they will enter through your mouth and enter to your digestive tract. And then they will attack where? They will attack to your intestine area, like this. This is how the Vibrio cholerae will attack your intestine villi. So what happened? Okay, we can have a look on this picture. So maybe I can draw you down better. You can imagine that this is your intestine villi along your small intestine, yeah, very long villi. And then what happened is the intestine villi, the cell of the intestine villi, actually they contains a lot of chloride ions. This is in the normal way. They contain a lot of chloride ion. So they will cause the intestine cell itself due to the high concentration of the chloride ion. They will appear as, we call as hypertonic. It's not hypertonic because low water potential. This one, low water potential. Why? Because that within the intestine cell, the chloride ions is higher concentration than water. Chloride ion more than water. Okay, more than the water molecule. So at here will be low water potential. So how about the outside? Outside here is we know as the intestine lumen here, which is the intestinal content in the intestinal lumen. So they usually have the very high water potential. Sorry, high water potential. So please remember that osmosis happens because water will always diffuse from the area of high water potential to the area of the low water potential. So the water will be moved in this way, diffuse inside. So this is how your intestine villi able to absorb the water molecule. So this is in the normal conditions. Normal. So how about in the abnormal conditions, which is when your intestinal villi is being infected by the Vibrio cholerae, is become in this way. This is your intestine villi, right? So the bacteria will secrete the toxin. So these toxins, they will cause the chloride ions losing out from the intestine villi. There will be a large amount of chloride ion leak out. So this will cause that intestine lumen itself contain a higher concentration of the chloride ion compared with inside the intestine villi. So now here will be lesser, outside will be more. So this will change our water potential, become that outside intestine lumens. What will happen? Outside intestine lumen, there will be higher chloride ions than the water. So now this is their turns become low water potential, because there's a large amount of chloride ion accumulate within the intestine lumen. Then how about inside the intestine cell? The intestine cell, they will become more water compared to the chloride ions. So this will cause the increasing in their water potential. So they will become high water potential. High water potential. So the water, they will go in this way, right? Because from high to low, they will go up. So this will cause the intestine cell losing large amount of water, because water go out. So the intestine cell, they will shrinking. They will undergo shrinking because they are losing water. And then your intestine lumen contents become too much water already. So until when they excrete out the feces, they will be excrete out too much water together with feces. So they will cause the watery diarrhea. This is known as watery diarrhea. Okay, so that's the story. So if you have a severe watery diarrhea, it will cause large amount of the water and iron being losing out from your body. So this is not good, okay? And this will cause the outcome, which is dehydration. This will cause, the dehydration, because you are losing large amount of water. So the patient with the watery diarrhea, they will suffer dehydration, and they are not able to control themselves due to the toxin released from the bacteria, in fact, on their intestine villi. So for the treatment, usually the patient will be treated by we call as the oral rehydration therapy. So what I mean by oral rehydration therapy, actually the patient will be given with the solutions, mixture with the salt and sugars, dissolve it. Yeah, this one. So they will be appearing this way. So here's an example of the cholera patients. They will be given oral rehydration therapy to replenish the fluid losing from their body. Okay, so remember that Vibrio cholerae. This is a commonly asked questions in the IGCSE. Okay, so the roots of the cholera is they will be ingested by the water and foods. So this is the process you need to know. Okay, produce the toxins, and then they will release the chloride ions, leak out. and then they will lower the water potentials. So the water able to move out from the cell into the intestine lumen by the osmosis. So they are losing water in form of watery feces. Okay, so this is a point that you should write in your exam. And then now, mechanical digestion. Okay, now we talk about mechanical digestion. Mechanical digestion, there's two places happen mechanical digestion. One is your teeth, another one is the stomach. Okay, chewing action of teeth, churning of the stomach, and the emulsification by the fats, by the bowel in the duodenum. Okay, so mechanical digestion also, they help to break down the food into a smaller piece. Just they do not alter the change of the chemical structure of the food molecule. They just change in their physical way. From larger piece becomes smaller piece. And then now we should talk about teeth. Okay, in human or in any kind of animal, actually we contain 4 types of teeth: incisor, canine, premolar, and molar. So incisor is at here. Incisors, you can have a look. This is an incisor. Okay, incisor will be the front teeth that we have around here. This is an incisor. This is an incisor. Okay, so what does incisor do? Incisor, they will help for the biting and cutting. Then how about the canine? Canine refer to the here. The teeth with a very sharp edge, like this. This is a canine. Can see? Teeth with very sharp edge. So what can I do? So canine, they will be help in the tearing, holding, and biting of the food. Especially tearing, okay? Especially tearing. So in the animal like the cats or tigers or lions, they have a very long and sharp canine, help them to tearing for the meat flesh. So you can see this canine. This is a canine. They have a very sharp pointed end of the teeth. So sharp. And then how about the, oh, sorry, I point wrong direction, is at here. Here, see, very sharp edge of the teeth. Help them for the tearing of the food. Then now we talk about the premolar and molar. So these 2 types of teeth, they have a very large and flat surface. The surface is very flat, like here. So flat. Okay, so they actually help in the chewing and the grinding of the food. Okay, so that's all. Yeah. So why they need to break down the food is they want to increase surface area of the food. See this point again. So they can expose more to the enzyme or the saliva. Okay, so please remember about the location of teeth. You can have a look on this. This is the front part will be incisor. Then the canine will be the adjacent to the incisor, and then followed by the premolar, and the backward. I mean the most backwards position of the teeth will be the molar. This is a molar. Here. They found in the most backward position of teeth. Usually there will be high incidence of the teeth. I mean the tooth decay happen on the molar because it's a most backward part. And then now we talk about the structure of the tooth itself. Okay, so the tooth actually is made from the calcium. Tooth is very hard. It's made from the calcium and the phosphate. So we can have a look on the structure of the tooth. So the tooth is made from the outer layer. The outermost layer we call as the animals. And then followed by the inner parts, which is a dentine. And then the pulp cavity. So inside the pulp cavity, they contains the blood vessel, nerve. Yeah, so this is a pulp cavity, which is a inner space within the teeth. And then they'll be surrounded by the semen. Here, this is semen. around here. And then the jawbone. Jawbone is just a bone, okay? Bone holding the teeth. Yeah, so those are structure of the teeth. And then you should know about what happened during the dental decays. Okay, so you can have a look on this. So every time when the tooth decay happens, they're actually starting from the animal parts. See, animals. Okay, so what will happen is whenever you don't brush your teeth every day, there will be the bacteria grow within your mouth. And then the bacteria grow is because that you didn't brush your teeth. There will be the remnant of foods left in your mouth cavity. So the bacteria, they're able to break down the foods, right? See, the food contain the sugars. So the bacteria will feed on the sugars. So they will undergo the aerobic respirations, okay, or the respirations. They will undergo the respirations, so they will turn into the acid. So this acid will dissolve the animal coating of the teeth. Because initially the teeth coating is very hard, right? So they will be slow, dissolved by the acid. Okay, so from the animals, and then they reach the inner part of dentines. And then remember that animals is always stronger and tougher compared to dentines. Dentine will be softer. Okay, they will be softer. So whenever the tooth decay reach the dentine, so it will cause the dentine dissolve very easily and quickly. So until very fast, they will reach the inner part of pulp cavity, until they will reach the pulp cavity. Because the pulp cavity, they contain what? They contains the nerve, right? Nerve and blood vessel. So they will cause a severe tooth pain, like this. Okay, so from the animals, they reach the dentines and the pulp cavity until they reach the nerve. Okay, and then when the bacteria live in the mouth, they are most harmless, but sometimes they will form the sticky film, which called as a plaque. This plaque, they just appear a very yellow, slight yellow coating cover your teeth surface. So if they will become, if the condition become more severe, they will form this tartar later. They will form this tartar. Okay, tartar, they will cause the structure become hardened. They will harden from tartar. So if they develop become tartar from the plaque, they'll be very hard to be treated. Okay, so this is the important that you should brush your teeth every day to prevent the bacteria growing, and they will produce a plaque and then lead to the tartars. Okay, so that's all about teeth. Teeth is very rare to come out in exam, but it come out before, just few years only. And then, yeah, so dental health, you should brush your teeth every day, and then you should use the fluoride toothpaste. So this fluoride toothpaste able help you to strengthen the animals to reduce damage from the acid. So actually the toothpaste, they have some alkaline property, help them to neutralize the acidic contents inside the mouth. Okay, so this is the enzyme action that you learned before, right? Last week. Okay, so I will go through it. That's all. Hydrochloric acid. And then remember that the stomach optimum temperature is not optimum temperature. It's the optimum pH is about pH 2 for the stomach. And then the bile, okay, the bile and the, okay, now we talk about the absorption of foods and the water. We continue of this part, this one, absorption of food and water. Assimilation, I haven't told you the assimilation. Sorry, let me have a look on the assimilation. Where is the assimilation? The liver assimilation. I think I missed out that assimilation. It's not stated in this. It's okay, later I tell you what is mean by assimilation. Okay, sorry. Okay, now we talk about the 7.5 absorption, neutral absorptions. So what is meant by absorptions? Absorption is whenever the foods already become very small and soluble, like glucose, amino acids, fatty acid glycerol, so they will enter into the intestinal villi, and then they will get into the bloodstream. Okay? Yeah. So usually the absorptions is occur in all part of the small intestine, especially in the ileum. Okay, so ileum is because it's very long. It's from the part of the small intestine. So the length of ileum is very long. So they contain a very highly folded surface with a lot of villi. So these villi, they will help them to increase the total surface area for the ileum to increase the rate of the nutrient absorptions. Okay, so you can see most of the absorption of water happen in the small intestine. Yeah. And then absorption, they usually take place in the ileum, which is the second part of the intestine. And then now we have a look on the villus. So this is a villus. You should know about the structure adaptation of the villus to help in the nutrient absorptions. So they contain very thin wall. The first structure is they have a very thin wall. We call it as one cell thick. Okay, because the thickness is just like the cell. One cell thick, very thin. So they allow the nutrients can be taken absorbed very quickly without them to travel such a long distance. They just require very short distance, then they're able to get into the blood capillary. Then secondly, they have a very large surface area. Due to they have a very highly folded, this one, highly folded villi structure. See, in wall, large surface area, and they are being surrounded by a network of blood capillary. They have the blood capillary. And then they have a lacteal. Okay, lacteal is the structure located at the center of the villi, which is they label as a brown color. This is a lacteal. This is a lacteal, right? Sorry, lacteal. So the nutrients will be absorbed into the intestinal villi, then they will enter into the blood capillary. Yeah, like this blood capillary. So after that, they will get into the liver for the assimilations. Okay, come. So after they enter into the villi, I mean the pathway. Okay. So this is a villi. So after the food, after the food enter into the villi, they will absorb into the blood capillary like this. They will absorb into the blood capillary. So from the blood capillary, they will enter into the livers. Okay, so there will be we call as a vein, a blood vessel connected between the small intestine and the liver. There will be the blood vessel. Okay, a small blood vessel connected from the intestine to the liver. So this blood vessel we call as hepatic portal vein. Because the nutrients, once they are being absorbed by the liver, I mean, once the nutrients is being absorbed by the intestine villi, they are still not able being used by the cell. So the nutrients, they need to travel from the intestinal villi, then they will enter into blood capillaries, and then they will get into the hepatic portal vein, and then travels to the liver. So the nutrients, once they reach the liver, they will help to process the nutrients so they can be able to use by the cell. So this is process called as assimilations. Okay, maybe I can show you the notes about this. We can have a look on this note first. This knot. Yeah, this knot. Okay, I think we should look about this knot first. Maybe I can describe to you on what is the, the important point in the chapter 7. I share my screen first. I already share the screen. So do you have any question to ask me currently? Eden, Suya? Okay, let me have a look on this. You can open this. This one, chapter 7, human nutrition, 学霸笔记. You open this one. Okay, this is very short and simple notes because I already put that the common mistake, which is the student do not understand or they feel that they are very hard to memorize. So I will put in this document. So for you to refer. So along the digestive tract, we have the goblet cell. So this is the goblet cell. Goblet cell are found in the digestive tract and also the respiratory tract. So what is function of goblet cell? Adam Suyang, what is the function of goblet cell? Want to have a guess?
Produce mucus.
Yes, correct. They produce a mucus in the digestive tract and also the respiratory tract. So the goblet cell, they look in this way, but in actual, during exam, they will not appear in this actual structure. They will appear in this structure like this. So you can see the goblet cell is actually very fat shells. Yeah, their structure is very fat. Okay, so yeah. So the goblet cells they will secrete the mucus. So the mucus will cover the intestine linings and also the respiratory tracts. So whenever we talk about the function of mucus in the respiratory tract, okay, this we'll learn in the chapter 11. Yeah, I think 11. They will help to trap the dust particle or air impurities. Okay, but how about in digestive tracts? So goblet cell, they will produce a mucus. So the mucus will cover the intestine lining. So the function of mucus is they are able to protect the intestine tract from being attacked by the bacteria or virus. Yeah, because the intestine tract is very, very fragile. Okay, so they can be easily attacked by any kind of bacteria or virus from the food content. So you need to protect. Usually it's for protections by the mucus. And also they will prevent the self-digestion by the enzyme such as pepsin in the stomach. Because you can imagine that your stomach content is so highly acidic. It's about pH 2. Very corrosive, right? So such a very corrosive stomach contents, they might be able to cause the corrosions, or they maybe can erode or corrode your stomach lining. So that's why your stomach lining, they need to cover with a thick layer of mucus to prevent the intestine, to prevent the stomach lining from being digested by the stomach. The enzyme, the highly acidic enzyme. So prevent the self-digestions, and also they help to prevent the physical damage by the foods passing through the intestine. So those are the purpose of the mucus. Okay, and then we continue. Yeah. Okay, about the process. Okay, the process you should know. This you should know very clearly. So whenever we talk about the process in the digestions, so ingestion is when you take in the food, you ingest the food, right? And then next step will be the digestions. So the digestion include the mechanical digestions and the chemical digestions. Chemical digestion is by the enzyme. Okay, mechanical digestion is by the chewing action of the teeth and the churning action of the stomach. And then the next step after the digestion, they will become from the, they will convert the large and insoluble nutrient molecule becomes small and soluble nutrient molecule. So they will be absorbed by the intestine villi. Yeah, this is intestine villi in the small intestine. Intestine villi in the small intestine. So after that, they will enter into the bloodstream, and then they will enter into the hepatic portal veins. See, so Then the neutral molecule, they will update by the liver to do the assimilations. Okay, assimilations. I'm not sure, do you still remember about the process assimilations? Assimilations, we refer to their definition is they help to process the neutral molecule become parts of the cell. Means the neutral molecule already become components of the cell. So at the end, they will become egestion. Means they will remove away the undigested. They will remove away the undigested material, become the feces. So this is no egestions, which is the feces. Okay, so this is for you to have a look. Where is the hepatic portal vein? You can see this hepatic portal vein, which is a blood vessel. You see, they are connected from the small intestines to the liver. This is a vein. Okay, so other vein, what are left gastric vein, mesenteric vein? You just forget it. You just need to know hepatic portal vein is enough already. Okay, so that's all. And then now we talk about, okay, this one. Whenever the exam asks you, state the usage of nutrients by the cell. Okay, so why we need to eat food every day? And we need to eat a balanced diet with the correct proportion of nutrients in every meal. Because we need to get these 3 type of macronutrients. Yeah, it's called as the main nutrients, which is the carbohydrates, proteins, and fats. So we need to take carbohydrates is because that we need to get the glucose. See, glucose. Because from the carbohydrate source, we're able to get glucose, right? Which is from the starch, it become maltose and then become glucose. So glucose, there will be, it's a called as a respiratory substrate. We call it a respiratory substrate. Respiratory substrate, this one. So they are used in the respiration, no matter it's aerobic or anaerobic. So the function is they want to release energy to the body cell. So these are the functions. Okay, so glucose they will provide as a main source of energy. Okay, so this is more important part. Glucose they will provide us with the main source of energy because they're able to use in the respirations to release energy to the body cell. So this is a point you should write in your exam. Then how about proteins? Okay, proteins is actually not the source of the energy. Protein is not source of energy, but proteins, they will help to tissue grow and repair and help us to build the muscle. So those require proteins. Okay, not only build muscle, but whenever your body want to produce hormones, enzyme, they also require the input of proteins. So usually protein is used by the body to build something. Then how about fat? Fat is an insulator. It's a great insulator. So the fat is found under our skin. We call it subcutaneous fats under our skin and also surround our internal organs. So this is because that fat able to provide a cushions. Okay, so these cushions able to protect our internal organs from being injured. And also fat will be the alternative source of energy. So this means what? It's not main source, but it's an alternative source. It's because that whenever your body, they fully deplete their carbohydrate source, what will happen? For example, if you didn't eat your meal for about 1 day, okay, you must feel very, 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 very hungry, right? So what will happen? So your body, they will burn the carbohydrate source in your store in your body, like the glycogens, right? The glycogens in the liver and muscle. So once they already finish deplete the source of glycogens in your liver and muscles, so they finish the source of they already finished the source of the carbohydrate, right? But they still need to get energy. So now they will start to burn the fats. They will burn the fats in your internal organs or under your skin to get the energy. So this is the sequence, okay, from the carbohydrate, fats, and proteins. Usually, your body will not will not break down the proteins to release energy. But if your body already deplete the fat source, means the fats around your, around your internal organs or under your skins already fully depleted because you still didn't get enough food intake, right? So now your body, they will use the proteins for the source of energy. So your body, they will start to digest the muscle tissue in order to get the energy. So this will this will be a very severe situations. And then hydrolysis, okay, or breaking down. Hydrolysis and breaking down it will be a same term, which is means digestions, okay, digestions. So you should know about the roots for these three neutral molecule digestions. So you can see for the proteins, they will undergo by the enzyme digestion, which is the pepsin, trypsins, and the erasins. So they will be absorbed into the blood capillary in the intestine villi. Okay, see, blood capillary. Carbohydrates will break down by the amylase and the maltase, then they will become glucose. So glucose will be absorbed into the blood capillary in the intestine villi. Then how about fats? Fats will be break down into the fatty acid and glycerol. This one, fatty acid and glycerol. So, however, fatty acid and glycerol are lipid. They are not water soluble, but they are lipid soluble. And then, please remember that fats are not able to dissolve in the water because the blood capillary, they contain blood, right? The blood is actually made out of water. So that's why the fats, if they enter into blood capillary, they are unable to dissolve. So that's why fat, they need to enter into lacteal in the villi, but not the blood capillary. This is, you should able to describe it. Enter into lacteal or enter into the blood capillary in the intestine villi. So once the fats enter into the lacteal in the villi, so they will enter into the lymphatic system, which is they will contain the lymph. Okay, they will contain the lymph. So another you need to know about, you realize that the neutral molecule able to get absorbed into intestine villi actually is the most simplest form, which is they are referred to the monomer. Okay, monomer like the amino acids. Amino acid is a monomer protein of the proteins, right? Glucose is also a monomer for the carbohydrates. And fatty acid and glycerol, they are also monomer of the fats. So will be the monomer form, which is the most simplest form. Then how about the disaccharide? The disaccharide like the lactose, sucrose, maltose, they are not able to absorb by the intestine villi because their size is too large already. Okay, and then please always remember that this trypsin, lipase, and amylase produced by the pancreas, they will transport by the pancreatic duct, enter the small intestine. So the digestion is happen at the small intestine, but not at the pancreas. Yeah. Okay, so the bowel two functions you already know, the bile, and then the structure of villi. Okay, structure of villi, you should know, lacteal, blood capillary, and the microvilli. Yeah. Okay, so this is a pathway. You can have a look. This one. So from the nutrients into the bloodstream, they will enter into the hepatic portal veins until they reach the liver. So from the liver, they will have undergo the process assimilations. This one assimilations, which is the nutrient molecule will become part of your body cell. Then how about the pathway for the lipid? Lipid is from the, once they enter into lacteals, so they will get into the lymphatic vessel, right? Lymphatic vessel. Lymphatic vessel is a part from your lymphatic system. So they will enter into the bloodstream and back to the hearts. Okay, so from the hearts, they have the aorta, right? The aorta is a large blood vessel. So the aorta, they will help to pump the blood to all of the body cell. Okay, to all the body cell. So the body cell able to get the fatty acid. So you can see assimilation of lipid happen in the body cell, but not in liver. Okay, but not in liver. So this is the difference, the pathway of the glucose amino acid and the lipid. Okay, so this is for you to have a look. You can see this picture. They show you about the nutrient transport pathway. You can see from the amino acids and the glucose, which is here. So they will get into the hepatic portal veins until they reach the liver for the assimilations. Okay, so then become part of the cell. But however, in the lacteals, they will become a limb, and then they will need to transport back into the hearts. You can see they need to transport back to the hearts. So from the hearts, they will transport to the all part body cell. So this means that the pathway for lipid, they do not undergo assimilation in the liver, but they will undergo assimilations of lipid in the body cell. Okay, so the function of liver. Okay, now we talk about function of liver. Whenever the question asks you about state down the function of liver, so this is an example of answer that you should write. Liver, they able to carry out carbohydrate metabolism. Means what? They able to convert the glucose become the glycogens. All glycogen become glucose. Actually, they are able to do the protein metabolism also. Okay, proteins metabolism. So this deamination is an example of proteins metabolism. So what is meant by deaminations? Maybe we have a look on this. This deamination. This one. Deaminations is involved in the removal of the amino group. It's not amino group. It's an amine group. Okay, amine group. This one. NH2. Okay, this is the amino acid structure. You can see the amino acid structure. You have this one, NH2 and the carboxyl group COH. Right, and then please always remember that the protein digestion process is like whenever your body have the excess amino acid. So the excess amino acid, I write here. I go right here. Oh, okay. Excess amino acid cannot store in the body. Okay, so this means that whenever your from the intestine, they produce a large amount of amino acid, right? And then maybe some part of the amino acid they will be used up by the body cell, but some part of amino acid they will still not being used up. So those amino acid they are not being used up, they will be excreted out from the body by the urea, in form of urine. They will not store in the body. It's not like the carbohydrates. If your body have excess carbohydrates, okay, excess glucose, right? For example, excess glucose. So the excess glucose able to store in the glycogen form, right? So they will be stored in the liver or the muscle. But this story is different for the proteins because whenever your body have the excess amino acid, they are unable to store in the body. So they will be need to undergo the deamination in the liver. So they will undergo the deaminations in the liver. Deamination is the removal of the amine group from the amino acids. Okay, so you can see that they are removed this NH2 group. So the NH2 group, they will become ammonia. So ammonia is toxic to the cell. Okay, it's a toxic to the cell. So ammonia, they will be converted into the urea. Okay, to urea. So now urea become non-toxic to the cell. So at the end, urea will be excreted out in form of urine away from the body. So this whole process, removal of the amine group, is known as the deaminations. So this deaminations happens in the liver, not in the kidney. Remember, not in the kidney. Some students they will thought this deamination happen in the kidney because that the urea is being excreted out by the urine, right? But it's not. Actually, it's happened in the liver first. Because the liver, they're able to convert the ammonia become urea. So only they will excrete out the urea by the kidney. Okay, so remember that excess amino acids are unable to store in the body. not like glucose, okay? So the deamination involved in the liver to convert the amine group become the urea, and then involved in the kidney help to excrete out the urea. That's all. Okay, and then you should know about the optimum pH range for the enzyme. So this also will be coming out in the questions in your paper 4. So you can see this is the optimum pH for the enzyme pepsin, which is pH 2. And then this is the optimum pH for the trypsin, which is about pH 8. Okay, so there will be 2 types of proteins that you learn, trypsin and pepsin. Okay, the watery diarrhea, I think that's all. Yeah, deamination. Okay, that's all for this chapter. And then now we talk about the past year question, which is this one. Let me share the screen again. Okay, you have a look on this. Chapter 7, important note paper 4. Okay, please open this document. Are you ready? Okay.
Yeah.
Okay, thank you. So you can see, describe what happens to the carbohydrates between injections and assimilations in the liver. So 8 marks they give you. So you should able to know how to describe. Yeah. So generally, okay, when you write about the absorptions happen in the small intestine, yes. And then you can write about the glucose. Okay, glucose will be absorbed into the villi. Actually, it's by 2 ways. One is simple diffusion or the active transport. This you will learn later. Okay, means that glucose are being uptake by the intestine villi, enter into blood capillary is by 2 ways, simple diffusion or the active transport. So after that, when we talk about the liver, assimilation happen in the liver, right? So the liver, they will do the assimilation for the carbohydrates. So means that the excess glucose, they will be converted into the glycogens by the insulin. Yeah, this is excess glucose. Glucose, they will convert into the glycogens. Okay, and then they will store in the liver and the muscle tissue. So we have the glycogen storage in the liver and muscle tissue, but majority of them will be in the liver. So this is for your extra information. Okay, what happens if too much glucose in your body? If there's too much glucose in your body, okay, until the storage of glycogen is become fully utilized. Because of this one, glycogen storage in the liver, there will be a limited capacity. Means that if you have about, maybe you have about 100 molecule of the glucose, right? You have only 50 molecule of glucose able to convert into the glycogens. Okay, 50 molecule of glucose able to convert into glycogen. Then how about another 50? Another 50 of the glucose, they are still exist in the blood. I mean, in your blood glucose level. So this means that this extra glucose, they will convert into the fats. This is for you to have a look. Means that if you have excessive amount of glucose until the glycogen storage already being fully utilized. So it means that the extra glucose unable to convert into glycogens anymore because the liver, they have a glycogen storage capacity. They have a limited glycogen storage capacity. So they will cause the excess glucose unable to convert become the glycogen and store in the liver. So this means that excess glucose, now they need to convert become fat. This one fat. unable to convert and store in form of glycogens. So what do? Is they will become fat. They will convert become fat. Acid glucose, they able to convert become fat. So the fat will store around our, around our skin, around our internal organs. And then, okay, important of calcium and the ions. So you just need to know that the iron help in the making of the red blood cell. Calcium is for the strong bone and teeth. Okay, so other than that, I think you already know. So assimilation, see this one, assimilation. Assimilation is they will help to move the digested foods. Okay, digested food is already small and soluble nutrient molecule directly enter into the cell, just like how glucose able to enter into the cell and get into the cellular aerobic respiration. So now the food already become part of the cell. And then for the lining of stomach, actually what form the lining of stomach? Okay, lining of stomach is actually formed by the, we call it as the cell, okay? So also we use the stem cell, this one. Stem cell, they able to undergo the mitosis. Stem cell are usually an unspecialized cell because the function of stem cell is they would just do the mitosis only to produce more and more cell. So after that, they will undergo the, we call as differentiation or specialization process, which is the stem cell, they will undergo the differentiation or specialization to become specific cell. It's not specific cell, sorry, it's a specialized cell, which able to carry out the specific functions. Like, for example, like the goblet cell. And then microvilli, they help to increase the surface area. And then if you don't have enough vitamin D, you'll be having this either rickets or weak bone. You will suffer those nutrient deficiency disease. And then this is the example of pancreatic cell, okay? So I hope you able to identify this structure. This structure is a mitochondria, yeah? It's a mitochondria. You can imagine that there's a highly folded membrane inside the mitochondria, like this. Like this. Really highly folded in the membrane. And then this is a nucleus. Nucleus is here. So this will be the endoplasmic reticulum around here. Okay, so this is for you to have a look. Balanced diet means that you should condense all type of nutrients in the correct or the right amount in order to maintain a good health. Okay, the mechanism of digestion, please talk about the mouth and the stomach. Yeah, that's all. And then okay, glycogen. Actually, what's mean by glycogens? Glycogens help to store carbohydrates in the liver. Actually, glycogens is a type of polysaccharide, right? It's a polysaccharide. It's a very important energy storage compound. So our body able to store extra glucose in form of glycogens. So the glycogen they will store. So whenever your body need the glucose input, so glycogen will be converted back into the glucose. Okay, so I think that's all for this one. Okay, yeah, I think let me discuss some question. I think we can start to discuss some question. Okay. Okay, this one for you to have a look. This is an intestine lumen. Lumen of intestine, okay? Within the intestine cell, they have the nucleus and also they have the rough endoplasmic reticulum, RER. And then they got the mitochondria. So number 8 is microvilli. It's a microvilli. So microvilli is they will increase the surface area to increase the diffusion of the digested food molecule, microvilli. Then how about the rough endoplasmic reticulum? So it's a site for the photosynthesis, for the protein synthesis. Okay, because rough endoplasmic reticulums, they will help to transport the proteins produced by the ribosome to the Golgi apparatus. And then mitochondria, they help in the aerobic respirations to release energy to supply to our body cell. And then about this, okay, this is an example of Vibrio cholerae, the image, yeah. So the ions, see, the ions, they are referred to the chloride ions. Okay, so this is the definition. Okay, you should mention about the keyword like chloride ions. Okay, they are diffused into the lumen of small intestine, and then they will cause the reductions in the water potential. So water able to move out of the intestine cell into the lumen. Remember this term, osmosis. Okay, whenever you write about the process involved in the water movement, please always remember to add osmosis inside. So they will call it dehydrations and watery diarrhea due to the loss of a large amount of the salts. Okay, this salt actually they are referred to the chloride ion because salt is made up of the sodium chloride and HCl. Okay, I think that's all. Okay, let me discuss your homework that you submit to me last week. Yeah, I think we can discuss our homework. Oh, sorry. Okay, this is the homework submitted by Ethan. Okay, I think I can discuss now. Yeah, we have some time. Okay, this all correct, yeah. So I think you already know about the structure of the every part of the organs. And then for this one, your answer is not really, I mean, it's not wrong, it's correct also, but you need to be more detailed. Okay, you need to be more detailed because how many marks they give you? 4 marks. Okay, if they give you 4 marks answer, I mean, if they give you 4 marks, you need to write at least 4 or 5 sentences. Okay, this one, this experiment is about you have the test tube A, B, C, right? And then what happened is the color indicator, which is they change from pink to colorless, indicate that the solutions change from the alkaline become acidic. See, from alkaline they will change become acidic. So for the test tube A, there will be no change occur, right? Because they are still remain in the alkaline due to the presence of bile. Due to the presence of bile, this one, bile. So they will still remain as alkaline. But in the B and C, they already change become the acidic because they change become colorless. But however, the C, they change faster compared to B. Why? Because the C, at least they contain the bile and the lipase. These two substance, which is lipase, enzyme, and the bile. So they help them to perform the fat digestion faster compared to the B, which is they only contain the lipase on it. Okay, so for your answer, the pH for B and C both decrease as they produce fatty acid. This is correct. I can give you 1 mark. And then in the test tube C, the presence of bile accelerate the production of the fatty acid by increasing the surface area for the bile. Okay, this one, increasing surface area for bile is not wrong. It's correct. Okay, it's correct. Maybe I can give 1 mark. So there's 2 marks out of 4 marks. But if you want to get a full mark, okay, because IGCSE you always score for the best, right? You want to score about A star, which is at least you need to get about 90 mark above. So you should add more. First, you can talk about what lipase do. Lipase, they help to digest or break down fat into fatty acid and glycerol. So the reason that the C change color faster than B, C change color from pink to colorless faster than B from the test tube B, because due to the presence of the lipase and the bulk and the bulk together. So this will cause the fatty acid produced in test tube C faster than B. And then you should state about the, we call it this one, why you're able to increase the surface area for the lipase. Why? Because that by able to perform emulsification, you should write this. By able to emulsify fat. Okay. Break down the large lipid droplet. Come small particle. So you continue. So you're able to increase surface area. Okay, continue with this sentence. So you're able to increase surface area for the lipid digestion by the lipase. So this will be the 4-mark answer. Increase the surface area for the lipid digestion by the lipase. So whenever if I'm the examiner, I will give you mark, right? So I will give you this one. Lipase digest into fatty acid glycerol, and then this one more, emulsify the fat, increase surface area for the lipase. Yeah. So this is a 4-mark answer. Okay, so just add on your point with the bioemulsification. And then about here, the purpose of test tube A, you see they give you 2 marks. Okay, if you give 2 marks, you need to at least write down about 2 sentences. Okay, so the complete question, the complete answer will be in this way. This show that bile does not digest fat chemically, right? Because that this one, test tube A, they contain the bile. So the bile, yeah, they're able to break down the lipid droplet, become smaller particle, but they are able to convert them become fatty acid. So the fat still remain as fat. Fat cannot become fatty acid glycerol because bile is not enzyme. Remember that bile is not enzyme. Because they are not enzyme. So they are unable to convert the fats, okay, into fatty acid glycerol. So that's why there's no production of fatty acid in the test tube A. Okay, so this proves that lipase enzyme, yeah, are required for the chemical digestion of fats to become fatty acid and glycerol. Okay, so you explain the reasons. Okay, which is bile is not enzyme. Okay, this one, yeah, this you're writing what? You are writing this graph. All lipase is denatured, but why you need to write here? Okay. I think this is not correct. This part. This part is correct. Until here it's not very deep.
It's like label.
Yes. Sorry?
It's not part of the graph.
Yeah, it's not part of the graph, but I mean in the actual way, you should write in this way. This like this, right? Oh, sorry. You just, because it's temperature, effect of temperature on the activity of lipase. Then they also what? Draw the label line and show the point where the lipase have been denatured. Oh, okay. Maybe you can write this way. I see. Like this, increasing, right, until reach the we call it as the optimum temperature point, and then suddenly they will decrease. Enough for you already. And then you can write here, you can draw like this temperature, this temperature and this enzyme activity. Okay, this one, adding a label line to show the point where the lipase has been denatured. Oh, okay. Actually, I know what I mean. Maybe you can draw in this way, like this. Like this, okay? This point. Because if you draw in this way, I worry the examiner might be confused. Your concept is correct. Just the answer, the way of answer, okay? And then we continue. Sorry, today we might end late a bit, but I hope I can discuss all of questions. Explain why lipase cannot be used to catalyze the breakdown of proteins, because that lipase enzyme can only use for specific substrate, which are fats. Yes, the shape of the active site of lipase is not complementary for protein. There's no product being formed. Okay, this is correct. I think I can give you at least 2 marks. 2 marks out of 3 marks, okay? Maybe you can continue add on with answer. You can say that proteins, because you already said that lipase is not complementary for the protein, right? So you can say that the proteins unable to fix into the active site of the lipase. Unable to fix. So whenever they are unable to fix, there will be no enzyme substrate complex being produced. You will write this enzyme substrate complex. I think I mentioned to you in chapter 5. Whenever you write the point, right, please remember to add on this enzyme substrate complex. Yeah, like this. And then this one, lipase enzyme can only be used for specific substrate which are fat. Actually, this is a bit weird. Sorry. This is a bit weird. Maybe you can write it this way. Okay. Lipase active site is very specific. The active site of the lipase enzyme are very specific, which they only allow substrate with complementary structure, which is what? Which is fat. Bind to its active site. That's all. See, your answer will be more detailed, right? Instead of you writing lipase can only be used for specific substrate, which are fat, and then what does it mean? So you mean specific. Specific means what? Specific. We call it as an enzyme specificity. They are referred to whenever the substrate they are able to bind to the active site of enzyme. So this we call as specific. So you should write about the process, which is how the substrate able to bind to the enzyme. See, bind to the active site. Use this term bind. Don't put like attached to the active site. No, please use this term bind. Okay, we continue. Sorry. Okay, so correct. The answer is correct. Okay, I think just like the way of explanation just improve, become more detailed. It's enough for you to score already. This that's all. Then how about this one? This one. This is we call it temperature, right? Temperature. And then what's your answer? As the temperature increase up optimum temperature, pepsin additivity will increase as small kinetic energy and successful particle collisions. Yes? Okay, then at here, since you write that you are increasing up as temperature increase, right? So you can talk about the kinetic energy increase. Yes, we talked already. So increased rate of collisions, but you can write about the between increased rate of collisions between enzyme and substrate. It will be more specific. Okay. Collision between enzyme and substrate. It will be more specific. This one. Because whenever I look at your answer, I'm thinking like examiner. Okay, what does it mean? Which one you refer? So your answer must be very, very specific. And then this one, pepsin optimum temperature at 40 degrees Celsius, higher activity as. Yeah, this is correct. Okay. Then pepsin denature temperature at 63 degrees Celsius. This is also correct. Yeah, denature. So there will be no more enzyme activity. Okay. Maybe you can write it this way. You see this one. Describe and explain. This is very common question coming out in the paper form. You need to describe and then you need to explain. Describe means that you need to describe the train of the graph. Describe the train of graph means what? Describe like this. Describe train of the graph. It's referred to. I think I put my answer below it. Okay. It's referred to when you look about this graph, right? The trend is, you can see they are increasing, right? Increasing from 10 degrees Celsius to 40 degrees Celsius until at the 40 degrees Celsius, they will reach a peak. And then after that, they will decrease significantly until they become zero. So you should describe the trend. Okay, so you can allocate about 2 marks or at least 3 marks. So how can you do? Okay, your answer, this one is, this is you will get the mark. Okay. This is you will get the mark. The no activity. Another 2 marks. And then I think you can get about 3 marks, okay? About 3 marks. Out of 6 marks. Okay. So if you want to write a complete answer, you should write this way, maybe you can describe that. You already write about the optimum temperature of the enzyme, right? With the figure. So after that, you write beyond the optimum temperature, what will happen? Beyond the optimum temperature, which is about 50 and 60, what will happen? So it will show the enzyme activity. There will be steeper decrease in the enzyme activity. See, steeper decrease from 40 degrees Celsius to 60 degrees Celsius. So until there will be no more activity. Yeah, this one you wrote already. No activity after maybe 62 degrees Celsius, 62 degrees Celsius. Okay, so this is your describe for the trend. Then now you need to explain, okay? So when you look at answer, explain is you are explaining in this way. This one. This is you explain it. And then this one you explain it. So when I want to add your answer, you can explain this way. See, you already explained that increased temperature, they will increase the kinetic energy of what? you can say increase kinetic energy of the substrate and the enzyme, right? Increase the kinetic energy of substrate and enzyme. So will be increase the frequency of collisions between substrate and enzyme. So you able to form more, okay? Increase number of formations of the enzyme substrate complex. Yeah. So you see this term enzyme substrate complex is very useful if you include in your answer, if the question is related to the enzyme activity. So what happened? Okay, so after when they beyond the temperature, beyond the optimal temperature, you cause the enzyme denaturation. Okay, so you should write about the reason why there's no activity here. This one. No activity because that temperature is too high. Too high temperature will break the bond. They will break the bond holding the enzyme. So whenever the enzyme lost their structure, okay, you can add on the active site of enzyme, they will be changed. Will change. Yeah, this is correct. Okay, so it will cause the lead to the denaturation. You should add this denaturation of the enzyme. Okay, so the substrate no longer complementary to the active site of enzyme. Yeah, so this is for the explain part, for the explain part. Okay. Then for this, explain why lactase is important for the nutrition of the young baby. Okay, you can say lactase is found in the milk. Okay, whenever you answer this question, I see explain why lactase is important for the young baby. Yes, the young baby, they drink milk, right? Milk contains lactose. So you should write the function of the lactase. Okay, you should write the function of lactase. So lactase do what? Lactase, they will help in the milk digestions. Lactase able to break down the lactose, become glucose and galactose, right? Yeah, you should include this. Okay, but this one I can give you 1 mark. And then this one, symptom of diarrhea, liquid defecation. Okay, usually not really described in this way. Yeah, okay, liquid defecations. Okay, interesting. But diarrhea we refer to what? You can say loss of the watery feces. Diarrhea refer to watery feces. Yeah, because our feces are excreted out in with a lot of water being excreted out also. Loss of watery feces, they will be correct term. And then how cholera cause diarrhea. Okay, so release the toxin in the body. Yeah, correct. And then cause small intestine have more water and leads to diarrhea. Okay, this is this that what I mean this sentence, cause small intestine have more water. Okay, this will be not really correct. You should write in this way. Just now I told you that you can write chloride ions. They are being leaked out from the intestine cell to the lumen, so they will reduce the water potential. Those points. Okay, I think that this is correct for you. Then liver, yeah, correct. Liver, gallbladder, small intestine. Okay, this will be a little bit hard for you. It's okay. This one I discuss with you next time. Okay, this is actually they are involved in the active transport. Yes, active transport you require the carrier proteins. You should write about carrier protein. Yeah, carrier protein. And the use of the energy against the conjugate gradient. Yes, just add more carrier protein enough. Because additive transport that happen in two situations, right? Together with the energy and the carrier protein. Okay, this one. Movement of chloride ions into pancreatic duct cause water move from the cell into the duct, help flow of liquid into the duct. Okay, from high to low through the semi-permeable membrane. But I think your answer is not really sufficient to get the full mark. Okay, if you want to get the full mark, you should write in this way. You should write in this way. Number 13. Okay. So this is number C. Okay, where's the answer? Sorry. Okay. Usually, when you want to describe about the movement of water by osmosis, it will be not really correct if you use this term. I mean, not really accurate. Maybe you can't get mark when you use this term in your exam. You refer to this term high concentration, low concentrations. In a better way, you can write higher water potential, lower water potential, right? You better write this way. So, due to presence of chloride ion in the pancreatic duct itself, okay, will reduce water potential. So this will cause water move by osmosis down the water potential gradient, which is from high water potential to area of the low water potential. Yes, through the partially permeable membrane. This is, you write it in the correct way. Partially or semi-permeable membrane. But if I'm the examiner, I think I'll give you 1 mark, okay? Because at least I saw the keyword osmosis. I saw the keyword of the semi-permeable membrane. Just like this is not really correct, this one. Okay, actually what this means is because that this CFTR, this one, CFTR proteins is a kind of carrier proteins found inside the pancreatic cell. So what they do is they will help to transport the active ions. They will transport the chloride ions out of the cell to the duct like this. Okay, they pump out. Okay, by the axial transport. So what will happen is, you can see, if the movement of chloride ions into the pancreatic duct cause the water to move from the cell into the duct, help to flow of liquid in the duct. So this means that whenever you pump the chloride ions outside to the pancreatic duct, right? So this will cause the accumulation of the chloride ions within the pancreatic ducts. So it will become low water potential. In the pancreatic duct itself. But inside the pancreatic duct cell, they will become high water potential. Right? So water will move from the pancreatic duct cell with a high water potential into inside the pancreatic duct with a low water potential due to the chloride ions. So this is the way that you should write, okay, by the osmosis. I think we stop until here, okay? I think time is already end. Oh, I'm sorry. Okay, we stop until here. I think we can discuss in next week. Okay, thank you, Ethan, Suyang, for your homework. And then do more exercise. You will become better. And yeah, okay, that's all for today. See you in next class. Okay, bye.
Time.